Hi, we're Team 2, and we're going to talk today about our project Fish and Chips, building a fluorescent system to image RNA fish microfluidic chips for diagnosis of HIV. I'm going to first walk through our need and approach using HIV as a case study, and then come back to some of the generalizable findings. HIV is a highly dangerous viral disease that in 2018 affected over 37 million people. It is a disease that remains high in prevalence, especially in developing countries, and is often difficult to treat. Testing for HIV is quite limited in poor and rural regions due to lack of convenience and affordability. It is also not strain-specific. Many recent HIV drug cocktails in developed areas are optimized for a certain strain of the virus, and it would be helpful to test for a given strain in developing areas. With these challenges in mind, the ideal HIV diagnostic for a low-resource area would be low cost, rapid, compact form factor. We believe that fluorescent in situ hybridization, or FISH, is a great way to do this. FISH is a highly specific way of binding fluorescent probes to mRNA. Using this technology, we can pinpoint viral RNA in cells down to the specific strain of the virus. Another advantage of FISH is that it can be optimized to be performed compactly on a chip. This has already been shown by the Isidore group at Penn. Here we see what a basic setup might look like on the left and what the image is that can be obtained on the right. Note that the cost per test is extremely low at only 5 cents. The main barrier for moving this into a point-of-care setting has been the expensive nature of the imaging setup. This is where we think we can innovate and adapt the technology to make it more applicable. Our proposed solution is an inexpensive fluorescent microscope to be used with a microfluidic FISH device for diagnosis of HIV at the point of care. Here are a couple pictures of our device and what it looks like. Even though we have only talked about HIV so far, this is a platform technology. By adjusting the FISH primers used, we could use our device as a cheap diagnostic for any viral disease, even COVID or the flu. Diagnostics like these are very needed at a time like this, and we think that this really increases the marketability of the device.
Our goals. Our main goal was to develop low-cost fluorescent imaging technology to perform an RNA FISH experiment on cells with our chip. How did we get there? To achieve our main goal, we had to combine cellular scale microscopy, fluorescent imaging, and a microfluidic device in an attempt to achieve classification accuracy, sensitivity, and specificity of about 95%. So onto our design process and the challenges that we had to overcome. First, we began by fabricating microfluidic devices that could be cheaply used to run RNA FISH assays based off of information from a paper published out of the Isidore lab. We then started developing an imaging system for the microfluidic device using a laser pointer lens, optical filter, and LED strips. A flow chart and a rough diagram of our initial system are laid out on the slide. Our first version of our imaging system involved using a press-fit box in conjunction with a laser pointer lens, optical filter, and LED strips. With the device, we could image cellular scale features. It could even count cells with MATLAB. We were also able to distinguish fluorescent material from non-fluorescent material. To get to that point, we had to test at least 10 different lenses and settle on the $7 laser point lens, seen in the picture in the bottom right corner. We iterated through several microscope chassis designs, first settling on a press-fit box with various holes for different purposes. we also tested 2 different LED setups in the first semester and tested at least 12 different optical filters in order to be able to include and exclude the appropriate wavelengths. Nonetheless, at the end of the first semester, we were unable to resolve cell size fluorescent features. We can only do either cell size imaging or fluorescent imaging, not both. This was due to the thickness of our filters being comparable to the working distances of the lens we were using. The lens had to be very close to our sample, as you can see in the picture on the right.
Due to our difficulty configuring the setup due to issues with the working distance as well as autofocus on the smartphone, we decided to pivot to another design. We found a resource online for a similar project that involved fluorescent imaging with a USB camera, and we decided to use this as a starting point for our next design. We had to make several changes to the design of the reference project in order to accommodate the microfluidic chip in a stable configuration, as well as within the working distance of our optical setup, since the cells we wanted to image are so small. This new setup was able to better accommodate fluorescent imaging due to the evenly spaced LED setup, as well as designing the parts so that the LEDs angled into the desired area of illumination in order to increase the intensity and decrease reflection. The LED color and power could be controlled through the user interface via an Arduino. Both the Arduino and the USB microphone would plug into a computer or laptop, and the images could be uploaded into the software on the laptop for analysis. Though we do not have images, when testing this device, we were able to see the 10 micrometer indications on the slide micrometer, and we were able to also see the fluorescence when using a sample of fluorescein. We plan to test the device further with fluorescent beads, as well as with cells fixed in our microfluidic device after spring break. Shown here is a comparison of our two designs. In our original design, the parts and casing were loose, which allowed for ambient light to be let in. Due to this, we were unable to detect significant fluorescence. This design also used a phone camera. It was difficult to focus on the same area over multiple trials due to the automatic focus feature of this camera. With the second design, the microfluidic chip was able to be placed at the appropriate working distance for the lens. The device would also be more easy to reproduce due to the greater cohesion and secure placement of parts. This secure placement also allowed for a tighter fit and less ambient light to be let in. The upgrade to the USB camera allowed for better focus and greater digital zoom. While exploring this new design, we also worked on a system to load a sample into the microfluidic device. This involved loading the desired sample into a syringe which connected to the microfluidic with a removable and watertight piping. We also developed the interface and general layout of our cell segmentation software. Unfortunately, we would only be able to develop more specific cell segmentation tools once we had sample images, as was planned after spring break. Taking the changes we made into account, we defined our final specifications as a digital USB-based optical setup with an Arduino-based LED setup. We would pass a 300 microliter sample in to be imaged on a 7.14 mm square imaging area. Overall, this device would cost about $100, though we expect that this price could be heavily reduced with mass production, as the materials we purchased were bought in singular or small quantities and were priced as such. Each microfluidic chip would cost $1, making this an overall affordable testing option. Down the line, with further developments, we would expect to reach the following testing stage. We would construct a dataset of 100 participants, equally split between HIV positive and HIV negative patients. Using their blood samples, we would run the FISH assay on our microfluidic chip and image each chip with our imaging device. The resulting images and their fluorescence levels would be split into testing and training sets. Various thresholds of fluorescence would be tested in order to determine the optimal threshold of fluorescence to classify an image as positive or negative. The number of true positive and false positive cases recorded at each fluorescence threshold would be used to construct an ROC plot. Using this, the optimal threshold would be determined. Following this, the threshold would be used on the held out set of testing data, and an overall classification accuracy could be determined.
Going forward, we would have evaluated our imaging system using 10 micrometer fluorescent beads and fluorescent cells. We planned on pursuing these next steps. First, we were going to image a nuclear stain of the cells with DAPI staining. Next, our plan was to undergo extensive testing to further validate our results. We planned on refining and optimizing our prototype to deliver a low-cost and effective fluorescent microscope. Then, we planned on developing a smartphone imaging app to visualize the viral detection and viral strain. Upon success of all these steps, we plan on taking these future directions. We planned on running the RNA FISH assay using green fluorescent labeled probes and imaging these cells with our device. The implementation of this assay would enable us to expand our tests to other applications. With the onset of the COVID-19 virus, we saw this low-cost, rapid test as a great opportunity to assist in testing underdeveloped regions. Some foreseeable challenges would have included signal noise. Our images would have likely contained background noise. To solve this, we plan on using computational analysis to assist with applying thresholds and algorithms in MATLAB to refine the signal. We also expected software compatibility to pose a challenge when integrating our GUI into other platforms such as mobile devices. However, we plan on running many iterations to achieve seamless integration. Finally, obtaining testing samples was a challenge we expected. To overcome this, we asked for the samples early on before break so they would be available after we came back. Although this was not possible, we have developed various technical and project management skills through senior design, and would like to thank Mike Magarachi, the Isidore Lab, and of course, the George H. Stevenson's Foundation Educational Laboratory and Biomaker Space of the Department of Bioengineering at Penn Engineering for the materials and space. Thank you for listening, and we're happy to answer any questions.